How can we use artificial intelligence to find new antibiotics? I'm incredibly pleased to welcome Dr. Jon Stokes. Jon is an assistant professor in the Department of Biochemistry and Biomedical Sciences at McMaster University. He received his PhD in antimicrobial chemical biology in 2016 from McMaster. The Stokes Lab leverages a mindful balance of experimental and computational approaches to discover and design antibacterial molecules with structural and functional novelty. A warm welcome to you, Jon. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And now, without further ado, I'd like to hand over to Jon to begin his presentation. Apologies for the delay, everyone, but we appear to be having some technical difficulties with Jon's audio, but hopefully...
Hello, everybody. It's a pleasure to be here speaking today. My name is Jon Stokes. I'm an assistant professor in the Department of Biochemistry and Biomedical Sciences at McMaster University. And today I'm going to be talking a little bit how we can use artificial intelligence to find new antibiotics. Sorry, I'm just sorting out my screens here. Give me a second. I apologize. Okay. So as as some of us may know, we've had antibiotics for the better part of about 100 years now. Alexander Fleming discovered penicillin quite by accident in the late 1920s. And in the ensuing years, we discovered a wide array of structural and functional classes of antibiotics. However, we can see, looking at this timeline, that the years in which we discovered our different structural and functional classes of antibiotics is not equidistant. It turns out that the overwhelming majority of our antibiotics that we currently use in clinical settings were discovered between the 1940s and the 1960s. This is the golden era of antibiotic discovery, and it's a period of time characterized by high productivity in discovering different structural and functional classes of antibiotics. And it turns out that most of these antibiotics, not all of them, but the majority, were discovered by looking for secondary metabolites produced by microbes that live in the soil. We can see, however, that come the middle of the 60s, early 60s, middle of the 60s, our ability to discover novel antibiotics by screening secondary metabolites produced by soil-dwelling microbes decreased. This is because we kept rediscovering the same antibiotics repeatedly. This is known as the dereplication problem. So in more recent decades, we turned to alternative approaches, medicinal chemistry, to modify the chemical scaffolds discovered through metabolite screening from microbes, as well as, even more recently than that, high throughput screening of synthetic chemical libraries. Unfortunately, even though we've been leveraging, medicinal chemistry and high throughput screening of, relatively large synthetic chemical libraries, we haven't been able to discover a fundamentally novel antibiotic class since the late 1980s, 1987. So currently we are in the aptly named discovery void, wherein we have not discovered any fundamentally novel antibiotics that have been deployed for clinical use. Now, it's interesting to think the discovery void in and of itself would not necessarily be a problem, right? Like we have, depending on how you look at it, maybe 15 or 20 distinct antibiotic classes shown here. However, the discovery void, of course, doesn't exist in a vacuum. And what accompanies the discovery void is the global dissemination of antibiotic resistance. And, there are isolates of various bacterial pathogens that cause illness in humans that are resistant to every antibiotic class we have on this slide. A recent report came out actually just a couple days ago. I don't remember the exact numbers, but roughly, 1.3, 1.5 million people a year die due to antibiotic resistant infections. And without immediate productivity, that number is expected to jump to, 8 to 10 million people per year by 2050. So what we need, given the inherent challenges of discovering new antibiotics, are, in my opinion, novel approaches to, enhance our ability to search chemical space efficiently and the search for novel molecules that are antibacterial and and inherently safe for clinical use. So one technological approach that could work to help us discover fundamentally novel antibiotics is the application of machine learning. And that's because these AI algorithms allow us to explore vast chemical spaces for novel chemical matter that is useful for whatever our application is. And what I'm showing here is, in a conventional wet lab setting, looking for novel molecules that are, let's say, antibacterial, we can test on the order of a couple million molecules in the laboratory. And we would expect, if we're using a naive chemical collection, roughly a 1% hit rate. So 1% of the molecules that we test will have the activity that we want. However, once we have, reasonable training data and we train a sufficiently robust AI architecture, we can easily look at, on the scale of billions of molecules. And again, depending on the ability of this model to generalize to novel chemical spaces relative to what it saw during training, we can expect, a roughly 10% hit rate or greater. Again, with many variables to consider, but this is a rough estimate of what one could expect. So that's why I think that the application of AI for novel antibiotic discovery is particularly useful. So I'm a big fan of, a couple different pathogens. This is one of them. Acinetobacter baumannii is a really, really challenging pathogen to develop antibiotics against. It's a nosocomial pathogen, so it's hospital acquired. So far as I know, it doesn't have a native ecological niche other than hospital settings, which makes it, to me, inherently interesting. It's gram-negative, meaning its cell surface is inherently impenetrable to a lot of antibiotics. And it has this really unique capability of acquiring DNA from its extracellular environment and just sort of keeping it. And given that Acinetobacter lives in hospital settings, clinical settings, a lot of that DNA that it happens to acquire and maintain encodes antibiotic resistance genes. So it's not uncommon to find Acinetobacter isolates that are resistant to most, if not all, clinically used antibiotics. And it poses a significant clinical burden because the people who end up getting Acinetobacter infections are oftentimes folks who are in hospital settings for a prolonged duration. So they're already immunocompromised and have comorbidities. So given that, Acinetobacter is a significant clinical challenge and it poses a unique drug discovery challenge, right, due to its, weird sort of lifestyle and propensity to accumulate antibiotic resistance genes, we figured, okay, this would be an interesting bug to go after in the context of novel antibiotic discovery. So today, I'm going to talk about two primary drug discovery model architectures, AI model architectures, both in the context of antibiotic discovery against Acinetobacter. So the first model type we're going to talk about briefly is called the discriminative model, and it satisfies the use case on the right. It says, I have pictures of cats and dogs, put all the cat pictures in one pile and all the dog pictures in another. And if we replace the word cat with antibiotic and the word dog with not antibiotic, this statement becomes, I have pictures of antibiotics and not antibiotics, put all the antibiotic pictures in one pile and all the not antibiotic pictures in another. On the other hand, we have a generative model, which satisfies a slightly different use case. Once we have a trained generative model, the use case is draw me a brand new picture of a cat that doesn't look like any of the cats you were trained on. So again, if we replace the word cat with antibiotic, it says draw me a brand new picture of an antibiotic that doesn't look like any of the antibiotics you were trained on. So we're going to talk about these two primary model architectures. And then right at the end, I'm going to talk about some brand new stuff that we've just started working on too. but we're not going to talk about it too much because it's not fully cooked yet. And it's just stuff that's emerging and sort of bubbling. Okay. So in the context of a discriminative model, right, if we want to, train an AI model that can predict new antibiotics, we have to train it on real world data. So the first thing that we do is we go into the laboratory, and this is actual lab data, right? So we had a library of 7,500 chemicals that we screened just for growth inhibition against Acinetobacter baumannii in a dish. And each dot is a different molecule. As we get closer to 0 on the vertical axis, this is growth inhibition. So every molecule here in blue, it was not a hit, and every molecule here in red we said is a hit. And this is defined using a 1 standard deviation hit cutoff. All the molecules in blue, non-hits, we labeled them with a 0. All the molecules in red, which are growth inhibitory according to this hit cutoff, we labeled with a 1. So now what we've done is we took this primary screening data and we turned this into a binary classification task. Molecules are either not active or active. And using this binarized data set, we can train a model, right? So what the model does is takes as input the structure of all the chemicals, converts these structures of the chemicals into high dimensional vectors. And then that high dimensional vector representation of every molecule is then used as an input layer into a standard feed forward neural network that then results in an output. And this is an output prediction score, right? So what we're doing, and there are many ways to do this, and I'm skimming over a lot of details. We can have a discussion, at another time about this. I'm turning structures of molecules into vectors and then turning that vector into a single number. And this number, the prediction score, is a number between 1 and 0, right? Because that's what we train the model on. It outputs numbers between 1 and 0. And this output is a reflection of whether the model thinks some input molecule is an antibiotic or not, right? So during training, what we're doing is we're training this model on, in our case, 7,500 structure property pairs. And based on what the model learned during training, we now have the opportunity to show that trained model brand new structures it has never seen before. And based on what it learned during training, it will convert those structures of brand new molecules it hasn't seen into a number between 0 and 1, depending on whether it thinks it's an antibiotic or not. So in this case, we have this molecule that we inputted into our model after it was trained, and it outputted a value of 0.15, which is close to 0. So the model is not confident that this compound is an antibiotic. And just to provide some sense of scale, after discriminative model is trained, using available infrastructure we have here in the laboratory, we can run predictions on the scale of 10 million per day, give or take. So once we trained our model, in this case on the 7,500 molecules, we wanted to run a very small scale prediction set. So we turned to the Drug Repurposing Hub, which is a repurposing library housed at the Broad Institute. At the time, it contained about 8,000 molecules. So we took all those molecules, ran predictions on them using our trained model. And what we observed was this plot here. So this is a rank order plot of all the molecules from the Drug Repurposing Hub based on their prediction score outputted by the model. So we can see that, a relatively small number of molecules have high prediction scores. We arbitrarily selected 0.2 as our prediction score threshold. So a small number have high prediction scores and a large number have low prediction scores. And this inherently fits with what we know about this library. We know that most molecules in the Drug Repurposing Hub are not antibiotics. And this is reflected in the prediction score distribution once we run predictions. What we then did is took all the molecules that had high prediction scores, meaning that the model was pretty sure that they could be antibacterial. And then what we did is we asked, out of all of these molecules, which ones are structurally distinct from every known antibiotic that we have, right? Because we're interested in structural and functional novelty when we look for new antibiotics. So it's not enough just to prioritize molecules based on prediction score. We have to also prioritize them based on structure. And it turns out that there was one molecule amongst these that had high prediction scores that was also structurally unique relative to every known antibiotic, and it was this compound here, which we named abicin. Abicin has a minimum inhibitory concentration of about 2 micrograms per milliliter against Acinetobacter. And when we test Acinetobacter against Abicin for bacterial cell viability, we observe that Abicin is, I'd say, modestly bactericidal against Acinetobacter in vitro, meaning it kills, roughly a log. Okay, so we had this molecule that was structurally novel and it worked in vitro. We then did an experiment to see what was the spectrum of activity. So we took this molecule and tested it against clinical isolate panels of Acinetobacter, carbapenem-resistant Enterobacteriaceae, Pseudomonas aeruginosa, and Staph aureus. Every dot on every plot is a different clinical isolate. And what we see on the vertical axis is growth, horizontal axis is increasing concentration of compound. And what we see is that as you increase the concentration of Abicin, we are able to inhibit the growth of every clinical isolate of Acinetobacter in our panel. However, when we look at our clinical panel of carbapenem-resistant Enterobacteriaceae, Pseudomonas, and Staph, this molecule doesn't work, suggesting that it was specific to Acinetobacter. And indeed, when we performed an experiment in parallel with this, testing Acinetobacter against a panel of human commensal isolates, it did not display activity. So we had this, narrow spectrum molecule that worked against Acinetobacter, and that was really interesting. And we said, well, what is the biological function that underlies this narrow spectrum activity? And through a series of experiments, I'm showing one here, we did a bunch of different experiments that all kind of pointed in this direction. We came to the conclusion that our molecule Abicin was inhibiting lipoprotein trafficking through a mechanism involving LOL-E. LOL-E is an essential protein that plays a role in the transport of lipoproteins from the inner membrane to the outer membrane in gram-negatives. What's really interesting is that LOL-E is universally conserved amongst gram-negatives, but the mechanism through which lipoprotein trafficking happens in Acinetobacter is slightly divergent from the mechanism through which lipoprotein trafficking happens in a lot of other gram-negatives. And I think that's what lends itself to having this narrow spectrum activity. This is the paper. It came out last year. It's super approachable. It's a really easy read. It's not written for computer scientists. It's written for, antibiotic microbiologists. So I encourage you to have a look. But there's an inherent limitation with discriminative models like I described, right? So discriminative models, like I said, can explore like maybe about 1 billion molecules before compute times become intractable, right? If you're running 1 billion predictions, right? You have a trained model and you run 1 billion predictions on some in silico chemical library. That's going to take you like a month, or at least it would take us about a month of continuous compute time. And after that, it takes too long. But we have to consider that like 1 billion molecules is not that many when we consider the totality of drug-like chemical space. And that's been hypothesized around 10 to the 60 molecules. And then I say, or whatever. And I say, or whatever, because 10 to the 60 might as well be infinity, right? And for reference, our solar system has 10 to the 57 atoms in it, right? So this means that the number of drug-like chemicals that could theoretically exist is equivalent to the number of atoms in 1,000 of our solar systems, right? So you can't comprehend that number. So the question then becomes more practical, like how can we begin to search the vastness of this drug-like chemical space for molecules that have the bioactivity and potential translational properties that we care about in the context of making a new clinical antibiotic. And given that a discriminator won't work, we have to turn to a generative model, right? So generative models for molecular design tasks are not new. They've been around for decades. The primary limitation of a lot of generative models that have previously existed is that they design molecules that look really, really pretty on paper, but are synthetically intractable, meaning we can't actually synthesize them, bring them into the lab and test them. But if we're interested in making a new drug, synthetic tractability is essential. And in the antibiotic space, giving the economic disincentives to making antibiotics, ideally making molecules that are synthetically tractable at scale for relatively low cost is sort of the gold standard that we're chasing. So to achieve this, we developed more recently, this was published earlier this year in Nature Machine Intelligence, we developed this algorithm called Synthmol. Synthmol is a generative AI algorithm for the de novo design of antibiotic design tasks. And what I'm going to do is paint a picture of generally how it works, then we'll dive into it a little bit more. Synthmol starts like before with the acquisition of a training data set and training a property predictor, right? This is stuff we already discussed. However, instead of showing the property predictor molecules from a pre-curated chemical library, right, we're going to design molecules to show to this predictor. And how Synthmol works is it doesn't design molecules atom by atom. Rather, it builds chemicals using a predefined set of molecular building blocks. So we have a set of 132,000 molecular building blocks, right? These are multi-atomic molecular fragments. And we have an associated set of 13 reactions. We know the structure of every building block. And across these 13 reactions, we know the synthetic compatibility of every building block with every other building block. So looking at every combination of 2 or 3 building blocks within this set defined by our 132,000 building blocks and 13 reactions, this gives us access to a synthetic space of about 30 billion molecules. This is a high-level representation of how it works. Basically, in 2 steps, it picks 2 building blocks that the model thinks will generate a molecule that is antibacterial. It then shows that molecule to the property predictor. And then the property predictor says whether it thinks it's antibacterial or not. We can then take these molecules that are predicted to be antibacterial, synthesize them for relatively inexpensively, and test them in the laboratory. So just like last time, we started with a chemical training data set. This time we used about 13,500 molecules, so we increased the size of our training data set. And this training data set was built across 3 distinct chemical libraries. What we're showing here in the bottom is a 2-dimensional representation of the chemical space that was explored amongst these 3 libraries. Black dots are every molecule that is labeled as an antibiotic from Chembl. We have molecules from library 1, 2, 3, shown in their respective colors. And then we have in red every hit molecule from every library. And what this shows us is that all of our hit molecules represent the totality of the chemical space explored amongst these 3 libraries. It doesn't say that all of our hit molecules live in one small region of chemical space, which would be problematic because it wouldn't allow our model to generalize very well to novel chemical spaces. So the only purpose of this plot is to show that our hits that are bioactive against Acinetobacter in the laboratory from our training data set explore the totality of the chemical space enabled by these 3 training data sets. Moreover, to help increase the robustness of this architecture, we didn't use one antibiotic prediction model. We used 3 antibiotic prediction models that are all slightly different in architecture. And we used 3 different models that are different in architecture because this helps helps by using 3 different model architectures. Each one is learning something slightly different about the structures of molecules in relation to their antibacterial activity. And again, this is a mechanism to help the model generalize and provide a little bit more confidence that when a molecule is given a strong prediction score, that strong prediction score is true. But this is just a detail that is not essential. One thing I want to mention is that we did not develop these 132,000 building blocks and 13 reactions. Rather, these come from the Enamine real space. This is a public repository of chemical building blocks and associated reactions. And the folks at Enamine were kind enough to give us full access to this to help us build Synthmol. So the inner workings of Synthmol are based on a Monte Carlo tree search, which is what I'm depicting here. So this is how the molecular generation process and the search of this ultra-large chemical space works. What we first do, right, level 0 is a root, so we can ignore this. At level 1, what we do is we take all 132,000 building blocks and show each one individually to our molecular property predictor that is trained on the 13,500 chemicals. What we then say is which building block has the highest prediction score based on our pre-trained property prediction model. In this case, we can see that it is this building block here. We then take this building block forward and we ask, what are all of the other molecular building blocks that are synthetically compatible with this highest scoring building block here? In this example, we can see that it is this building block that is most likely to be antibacterial based on our pre-trained property predictor. And as such, we move forward through this node in our tree. We can then see that these two building blocks are synthetically compatible through reaction 11, whatever that is. We can then in silico synthesize the resultant molecule, show the full molecule to our property predictor, and then get an output score from that property predictor. In this case, we can see the output score is 0.9, which is very high, indicating that the model thinks that this molecule is antibacterial. And as such, this 0.9 high prediction score from our property predictor is then back propagated, and the nodes that correspond to the two building blocks that made this molecule are updated accordingly, meaning that the individual scores for this building block and this building block are increased. Right. Conversely, if the prediction score for this molecule is very, very low, that low prediction score would be back propagated, and the nodes corresponding to the associated building blocks would have a score that is decreased. Now, you can imagine if you run this over and over and over again, we're going to have building blocks that make good antibiotics have scores that increase over time, and building blocks that make poor antibiotics are going to decrease over time. So what we're doing as we're updating all of the weights, all of the scores of each node in our tree, is we're learning how to navigate this large combinatorial chemical space in order to find building blocks, which when put together, make high scoring molecules. We did this process 20,000 times, and we were able to synthesize 58 molecules, which we prioritized for wet lab testing. And of the 58 molecules that we synthesized and brought into the lab for testing, 6 of them were bioactive when we tested them against Acinetobacter, MRSA, as well as other escape pathogens. So we had roughly a 10% hit rate. And for reference, the inherent hit rate of this chemical space is less than 1%. So we had at least an order of magnitude higher hit rate relative to random screening of this same chemical space. And that suggests that this whole Monte Carlo tree search-based synth-mole architecture worked. This is the associated paper. Again, it's not a particularly challenging read. It came out earlier this year. I encourage you to read it if you have an opportunity. And here's what we're working on now. I have 2 really small things we're working on, and then we'll be out of here. One thing we've already done is we've improved the synth-mole architecture. So synth-mole, the original version, was built using a Monte Carlo tree search-based method through which various molecular building blocks were prioritized to make molecules. However, that is slightly suboptimal because MCTS can't generalize its learning across similar building block structures, right? MCTS does not learn the chemical structures of individual building blocks during the molecular generation process. All it knows is to travel to different nodes, right? A building block happens to live in a node, but it doesn't know the structure of that building block. Therefore, we have optimized synth-mole by employing a deep neural network instead of MCTS to score and select building blocks. And this deep reinforcement learning architecture learns the relationship between the building block structures themselves and the resulting product's antibiotic score. And that means the deep reinforcement learning architecture can generalize better across structurally related building blocks. So in this example that we're currently putting together right now, we had a Staph aureus training data set, and we were trying to multi-property optimize for both Staph aureus data and solubility. So we have the molecular generation phase, the building block selection phase, being driven by a deep neural network. So what happens is it, much like before, it selects two high-scoring building blocks, puts them together, and then what it does is this architecture takes that molecule, shows it to both the Staph aureus prediction model and the solubility prediction model, gets two outputs for both. And then based on the aggregation of those two outputs across these two models, it can then update the weights of the reinforcement learning model such that in future rollouts, future rounds of generation, the reinforcement learning model gets better and better at selecting building blocks that result in molecules that are both Staph active and soluble. And indeed, when we run a small scale experiment using either our deep reinforcement learning model, which here we've called RL-ChemProp, or MCTS across 10,000 rollouts, looking for molecules that are both predicted to be Staph active and soluble, our deep reinforcement learning model is capable of generating about 1,300 molecules that satisfy both properties, whereas our prior MCTS model can only generate about 350. We're currently in the process of testing our deep reinforcement learning model outputs in the laboratory to validate their in vitro efficacy. We already have a couple molecules that work in vitro, and we are progressing those to in vivo studies as we speak. The last thing that we're particularly interested in and actively working on now is the application of AI methods to help predict the mechanism of action of structurally novel molecules. So in collaboration with Regina Barzilai from MIT, we've employed a diffusion model, which is a type of AI model that has been commonly used recently for the modeling of small molecule protein interactions. And we have leveraged a diffusion model to help us predict the protein target of a structurally and functionally novel small molecule. To my knowledge, I'm not aware of any other groups that have leveraged diffusion models to help identify the binding site of a novel small molecule to a potential protein target. So we're super excited about this paper, and we're just wrapping it up currently as we speak. And the goal is to, continue to grow this work to develop a more robust tool that folks can use to help predict the protein targets, if it is a protein target, of novel small molecules that you're working on for whatever therapeutic application. So we're just wrapping this story up as well. So that's my time. I don't do very much work. I answer emails for a living. This is the group that does everything. Huge shout out to Denise, who was driving the diffusion model project, and Gary, who is driving the SynthMol project. And of course, everybody in my lab is just incredible. So thank you so much for your attention, and I'll take any questions you might have.
Thank you very much, Jon, for that fascinating talk. Before we continue, I just want to confirm you are able to hear me all fine. Are you able to hear me, Jon? It seems Jon is having some technical difficulties, but hopefully we'll be able to get back on track with the Q&A in just a second. Are you able to hear me now, Jon? Can you hear me, Jon?
I can hear you now. Apologies.
Excellent, fantastic. No worries at all. I'm glad we got there in the end. Firstly, thank you so much for that incredibly fascinating talk. We really do appreciate you sharing your expertise and insights with us today. As of all the other talks in the event, we're likely going to have lots of questions from the audience, and I can see that we have received some questions already. So I'll move straight onto the Q&A section now. So Jon, your first question today is, how can you maximize the ability of a model to generalize to novel chemical space?
Yeah, so it's a perfect question, and one that we grapple with all the time, because that's like the fundamental limitation. So there's this really interesting tension that exists, right? So let's say everything in my screen here is like a chemical space in which we're interested in working, right? And if my training data set is defined by this block over here, and I want to run predictions on a chemical space over here, this is a really long distance, right? So my model is unlikely to be able to generalize this far, right? If I train a model on data up here and then make it predict down here, there's a good chance that whatever prediction scores I get from the data down here, the model is just going to be guessing, right? Just full on guessing, because it doesn't know. It's like, I'm a biochemist. It's like if you put me in like a 4th century literature class, I wouldn't know what was going on, right? But if you put me in a botany class, I might be able to guess. Okay. So the best method through which you can get a model to generalize is to increase the chemical space that is represented during training. That's the obvious way. Oftentimes, you don't necessarily have that luxury because you have a predefined set of chemicals that you have in your laboratory that you have available to work with, right? So in this case, you have to leverage, unique AI model architectures that may be inherently better at generalizing to the chemical space to which you want to generalize. And this is often where more sophisticated neural approaches come into play, based on, stuff we've published as well as stuff that we just sort of anecdotally observe in the laboratory. Different model architectures, simpler ones and more sophisticated ones, are inherently better and worse at generalizing to novel chemical spaces. So this boils down a lot of the times to guess and check. You take your training data set, you make it as diverse as you can, and then you train a whole bunch of different model architectures and ask, which one do I think is best at generalizing in the chemical space that I want to work in? And then you just pick the one that is the best, right? If there's a better way to do it, we haven't found it yet. That's how we do it. And it works. It works good enough, right?
That's excellent. Thank you so much, Jon, for all the insights and your fascinating descriptions. That's an excellent answer. Now, we had a question that came in quite early on during your presentation from a member of the audience. So it was, what are the name of the programs that you use to make this kind of AI prediction? I know that you mentioned Synthmol, but are there any others that you utilize in your research at all?
Yeah, so like Synthmol, we developed Synthmol. So and that, like, we do, again, I don't do anything. I answer emails all day, but like that's all coded like in Python, using like available stuff. And all of that is freely available on GitHub too. Everything I discussed today, you can go on to GitHub, like you can go to the papers, go to the bottom, say, where's the code, click the link, and we have everything, our training data, everything freely available. But our molecular property predictor, the one that we often use most frequently, and again, tends to give us the best prediction accuracy when we try to force these models to generalize, is a message passing neural network library called Chemprop, CHEMPROP. So if you go onto Google and type in Chemprop GitHub, this is a model architecture that has been refined over the past probably like 7 or 8 years. Again, Chemprop comes out of a good friend of mine, Regina Barzilai, her lab out of MIT, largely in collaboration with Tommy Yakula, I believe, too. He's another computer science professor at MIT. But this is a very, very highly engineered system that is, relatively user-friendly for anybody to pick up and apply to their own training data set for their own drug discovery task. So Chemprop, CHEMPROP on GitHub, you'll find it. And then if you type in Synthmol GitHub, you'll find it. It's all there, everything.
Fantastic. Thank you so much, Jon. We have time for a few more questions today before we conclude the session. And another question that we had come in from the audience during your discussion was, did you incorporate explainable AI to explain how the model is learning the structurally important components?
That's a really, really good question. And and the stuff I discussed today, we did not. I'll tell you, we're working on a project now through which we're applying a couple different explainability methods to begin to understand why a model is making the decision it's making. And there are a couple ways that one can do this. And here's how I like to think of this problem. It's like we have a molecule. Here it is. It's a brand new molecule, and it doesn't look like any other antibiotic ever, but the model's convinced it's an antibiotic. It's like, tell me what features of this molecule are giving you the idea that this is an antibiotic. Like, because me as the quote unquote expert, I can't see why you picked this, right? And though like we're trying a bunch of different methods and seeing just like which one sort of makes the most sense for our applications. And I think that's a really, really good question, because when it comes to leveraging the outputs of these AI models to make like medicinal chemistry decisions, for example, like a synthetic chemist would want to know, like, okay, your prediction is right. Okay, what parts of this molecule do I need to keep in order to keep the correct prediction? But what parts do I have to play with in order to optimize for the properties that are suboptimal currently, right? And that this is still like, it's relatively early days for this. And there's a lot of sort of activity at this like explainability chemical property interface. But in my opinion, we're not quite we don't really know what we're doing just yet. I think it's going to be a couple more years, but it's a perfect question.
That's fascinating to hear. Thank you so much, Jon. We do have time for perhaps one final question, and I know that we do have a lot of questions that we may not be able to address, but they will be addressed offline as soon as possible. So, Jon, the final question is, how do machine learning models differentiate between known and novel chemical spaces when searching for new antibiotics?
How does can say that again? Sorry, say that again.
How do machine learning models differentiate between known and novel chemical spaces when searching for new antibiotics?
Oh, I see what you're saying. Okay, so how do they differentiate between the chemical spaces? I think they don't explicitly differentiate. So, like, I want to go back to like my hand analogy, because that's all I got right now. I wish I used to have a whiteboard up here, but I pulled it down and put my flag up. I don't know why I did that. Terrible decision. But like the model, you have to think about like this. The data that you show your model is like the whole universe of what that model knows, right? Because like by definition, you've given it a book, and it's like, this is the whole book. This is the entire universe that you are going to understand, right? And if the training data set, that like book is up here, what the model learns is like everything that's in here is the entirety of the universe, right? So then by definition, if you start to show it new molecules that live like just outside here, it's probably going to be able to generalize pretty good, because it doesn't have to stretch its imagination, if you want to think about it like that, so far, because it's like next door neighbors. It's like right here, right? So in the case where you have a model that's trained on this chemical space making predictions here, it's probably going to be okay. It's going to be guessing a little bit, but not everything it outputs is going to be a guess, right? Now, if you start to show it stuff that's out here, like the model doesn't have any idea about this chemical space over here, because this is very far outside of its universe, what you told it during training, right? So again, it goes back to this idea, like the model doesn't, necessarily know that it's out of distribution, but it's seeing these chemicals and like when it's trying to interpret these molecules based on what it learned during training, it's saying like, this is unrecognizable to me. I don't know what to do. And what's going to happen is that output score, and we've seen this empirically, is either going to go and asymptotically approach 1 or asymptotically approach 0. Right. And we've shown models that are trained on one chemical space, brand new compounds that exist in like really weird chemical spaces. And it'll be like, I'm certain this is an antibiotic. And we get an output score of like 0.999999. Even though we, as like a human, go like, oh, that's totally wrong. Like, you really, really don't know what you're doing here. right? And that's actually a pretty good test. If you take a chemical space that is very, very far at a distribution relative to training, you'll end up getting just like these wild prediction scores that don't make sense. So it's not the model, like the model doesn't know that it's making predictions out of distribution. It just so happens that when you show it chemicals that you know are out of distribution, the model does just like weird stuff.
Excellent. Thank you so much, Jon, and thank you for clarifying that so perfectly. But unfortunately, that is all that we have time for today, but we really do appreciate you joining us to share your expertise and for answering all those questions.
Yeah. And if there's others, just send me an email. Like, search my name on Google, find my email, and I'll get back to you.
Fantastic. As mentioned, any questions that we didn't have time to get to will be sent to the speaker directly after the event. And as with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. So that just leaves me to say thank you once again, Jon, and to everyone in the audience, we'll be back soon with the next talk.
Thanks, everybody.